Hi, everyone. My name is Samantha. Thank you for coming to this stream. I'm very excited. This is my first one. I'm a student streamer, as you can probably tell. And today I will be leading a review in chemistry for AP Biology. The biggest thing you can do to prepare for the AP exam is literally to study and to practice and to do your homework, do the labs. The more you understand concepts, the better you'll do, because you aren't going to be tested about the same specific thing every year. It's going to be hidden and it's going to be tricky. So I'm a senior this year. I took AP Bio last year as a junior. I loved it, my favorite class ever. I scored a 4. I'm not the great at testing, but I was really happy with my 4. So the biggest thing with the exam is that they are going to test how well you understand the big picture and how well you can apply these concepts to different problems. So they'll present a situation, they'll give you all of the info you need, and you just kind of have to fill in the blanks. So that's why I think doing a stream like this, like knowing your chemistry, it's kind of like knowing the theory. You know, when you know that, things just click easier. And so this review probably won't be learning anything new. Maybe in the second half when we start to apply it to some biology concepts that I know Fiveable is currently teaching. So we'll talk about some biological macromolecules if you're keeping up to date with the streams. But if I can reinforce some stuff and if stuff can click later on because we talked about this, that would be the goal. All right, so as always, follow Think Fiveable on any social media. Keep up to date with our really cool memes. Instagram is full of them. YouTube, Twitter, great way to keep up with the current streams and such. Also, in this stream, so I already did kind of my introduction and advice. That's kind of my overview. You'll get a lot more advice out of me throughout the year as I do more streams because I have a lot of opinions. I made a lot of mistakes and I learned from them. And then we're going to do some chemistry review. So terms, ideas, concepts, all very basic. And then we're going to start to apply it to biology. All right, so the very first thing you need to know for a chemistry class ever is the atomic structure. So as you can see on the right here, I have this very lovely atom. And in the atom, you have your protons, your neutrons, and your electrons. So your protons have the positive charge, the atomic weight of 1. They're found in the nucleus. The neutrons don't have a charge, neutral, if you will. Also found in the nucleus, also has that weight of 1 to balance the nucleus out, if you will. And then you have your electrons that are negatively charged and found on the outer shell or outer shells. And because you guys are super smart and have taken good honors and or AP chemistry classes, you know all about valence electrons, the electrons that are on the outermost shell, and that will come into play a lot when we talk about different kinds of bonds. All right, so our first kind, ionic bonds. So ionic bonds happen when an atom will gain or lose an electron accordingly to achieve stability. So I have these very nice diagrams down here, and I'm a very visual person, and I think science especially is a very, very visual course. So I like to include a lot of diagrams as well. Oh shoot, sorry guys. All right, so as you can see in the diagrams below, this sodium has one extra valence, or one valence electron. It doesn't have an extra one. So 1 on its outer shell, and this chlorine has 7 on its outer shell when it can hold 8. So as seen in this diagram, it gives its extra 1 to the chlorine, and it completes the valence electron outer shell for the chlorine, gets rid of it for the sodium, and they are stable and they're happy. And you have your classic NaCl, sodium chloride, you have your table salt. I have some vocab listed in here as well. Ions, electrically charged particles that result when an atom gains or loses electrons. Cations and anions, so positively and negatively charged, respectively. I'm sure you guys are all super familiar. So next we have covalent bonds, which are really, really important in biology. You're going to see a lot of covalent bonds within some of these macromolecules. You're going to start to kind of see why these are important to biochemistry, to life. I feel like a lot of times in chemistry classes, they just kind of explain everything. They don't really tell you why it's important or where you see it, or at least my chem teacher never did. So it'll be really cool when you see why covalent bonds are so important. But covalent bonds, as you guys know, covalent bonds are when two atoms achieve stability by sharing an electron. And so these can occur in single, double, or triple bonds, depending on the availability of electrons from the two atoms that are sharing. Covalent bonds are known for being super, super strong. So lots and lots of energy is needed to break them. And so then you have electronegativity, which is super important in covalent bonds because those are going to affect whether or not the bonds are polar or nonpolar. And that is another concept that will come up a lot throughout AP Bio because, again, like I said earlier, like you're going to see why it is important that certain bonds are and aren't polar or nonpolar, whatever. So again, I have some diagrams. So I'm just going to walk through them really quick. So, you have your hydrogen, and then you have your hydrogen that are sharing your 2 electrons. So it becomes H2, and that's really stable. They like it that way. And then here you have your fluorine, and this is still just a single bond. And a double bond would be when they're sharing 2 electrons between the 2 of them. That would be when they're sharing 4 between, like, total. And 6 would be a triple bond. And those are mostly found between carbon molecules. Oh, no, wait, I'm sorry. I just misspoke. Triple bonds are usually found within nitrogen. You'll see a lot of double bonds between carbon, and double bonds do become important down the road, so make sure that you do understand that. And so electronegativity, I did kind of skip over that. So in case you are unfamiliar, electronegativity is if one atom has a stronger force than another one that it is sharing with, it'll tug on the electron a little bit more, making the atom's charge partially negative. So it's not fully negative because it doesn't fully have the atom, but it also isn't neutral because it pulls on it a little bit more. So therefore, the atom that is more electronegative pulls on the electron a little bit more, is partially negative. The one that is less electronegative becomes partially positive. If this is occurring, that means that it is polar. Best example of a polar molecule. Can you guys fill in the blank? I'm sure you can. No, just checking that we all know. Can you say it in the chat? I want to make sure you guys are still with me. No? Okay. And you have water. Okay. And nonpolar is when neither of them are more electronegative than the other. So because it's nonpolar, there's no poles. Okay, so finally, our last kind of bond that we are going to talk about today are hydrogen bonds. So hydrogen bonds are, and this is right here is when polarity becomes important. So a bond between a strongly electronegative atom and a hydrogen atom. So because water, H2O, is polar, the oxygen pulls on the electron a little bit more, becoming partially negative. The hydrogen is then partially positive, the hydrogen atom within the H2O molecule. And so the partially positive charge in the hydrogen portion of the molecule and the partially negative aspect of the oxygen atom in the molecule are attracted to each other because, opposites attract. And so these bonds are not as strong as covalent bonds because they are solely just an attraction. So they're not as strong, but they are important, equally important, actually. So hydrogen bonds, they are important to know because they have a high heat capacity or a heat of vaporization. So it takes a lot of heat before they break. and they are very cohesive. So this is why water is super, super cohesive. This is why water sticks together and stays together almost always. Like you could have, your drop of water and then put another drop right next to it and they'll stick together. And it's because of this right here that these molecules can bond together. It's because of the hydrogen bonds. Okay, and so before we move into how this applies to biological macromolecules more specifically, I just want to go over some extra vocabulary that is more chemistry than it is biology, but you'll see either within this unit or more so within cellular respiration and photosynthesis, because that has a huge biochem aspect in it as well. So you have hydrolysis, which is the chemical breakdown of a molecule and or bond due to a reaction with water. So it basically and you'll get a visual of this in a little bit. I did include it, but essentially if a bond is broken, you would need to add water because it needs those extra hydrogen and oxygen atoms to complete that separation. The opposite of that is not on this slide would be dehydration synthesis, where the bond forms and water is released. So those 2 hydrogen and that 1 oxygen atoms will be expelled and a bond will form. And that will be important as well. And we will see that will become visual. Hydrophobic and hydrophilic describes whether a molecule is repelled from water, which is phobic. If phobic, root means you're afraid. Hydro meaning water. And attracted, philic meaning loving, hydro meaning water. Hydrophilic means it is attracted to water. This becomes very important, which you'll see in a few slides as well. Catabolic reaction is the breaking down of a complex molecule into a simpler one, and it releases energy. That's really all you need to know for right now. That's not going to come up in today's stream. That may or may not come up down the road. I remember seeing that word used a lot when I took AP Bio, and I was just like never really sure what it was. So I just threw that in there just to have. And then the last one I included on this slide was oxidation and reduction, or redox reaction. So any reaction that involves a transfer of electrons. So one will gain an electron and one will lose an electron. Reduction is when you gain an electron, and Leo is when you lose, oxidation is when you lose. I was just thinking of an anagram that they taught us, and I'm pretty sure I'm correct on that. I'm sure I'm correct on that. But that will come up more so in cellular respiration and photosynthesis. All right, so this is a broad overview. These are your 4 big biological macromolecules. Actually, wait, before I go on, is everybody good on everything I just said? If you have a good chem background, you probably will be, because that should just be review. Yes, everybody's good? All right, just want to make sure I'm not moving too fast or anything. I wanted to give you a chance to ask questions. Perfect. Everybody's with me. All right. So biological macromolecules. These are your 4 main macromolecules. You have your carbohydrates, your lipids, your proteins, and your nucleic acids. And I put in basic forms or basic diagrams for each of them. And I will explain carbohydrates and lipids in more detail here in a second. I didn't want to explain proteins in detail because that turns into a whole other unit. So I will be doing that down the road, maybe in a few weeks, in a few months, but not today because we don't have to get into that. But essentially, these bonds become really, really important as proteins are determining their structure. So long story short, you have a polypeptide chain of amino acids, and the way they react with each other, the way that they're bonded, actually does determine the function and the identity, if you will, of that protein, which is why that's really important. Nucleic acids do have bonds within them. Obviously, that's really important because they are macromolecules, so they do have multiple parts to them. For today's video, I just didn't want to get that complicated with explaining the chemistry behind that, just because it's a lot, and it's a lot of stuff that wasn't mentioned, but you'll get there and you'll need to know, and you'll know everything that you'll need to know. So I'm not worried about it. You guys are all super smart. But for today's video, we will not be going super in depth about that. But in terms of carbohydrates and lipids, we're going to talk about some cool stuff and how this is where we'll learn where things in chem do actually matter and do keep us alive. So, all right, so let's move on to carbohydrates. So carbs or carbohydrates are made up of carbon, hydrogen, and oxygen. And the reason I have CHO in parentheses is because how I remember what is what. I'm going to flip back really quick. So I just remember CHO, CHON, CHOMP, which is something that I learned from an Amoeba Sisters video. But essentially it's just CHO meaning carbon, hydrogen, oxygen, although lipids do occasionally have phosphorus, but we'll get there as well. Proteins have carbon, hydrogen, oxygen, and oh shoot, wait, I messed this up. This has, do you know what? Forget it. We'll talk about that in a later video, but it's a good way to remember it. But that's why I have CHO there. I forgot to delete it. The important thing to remember with carbohydrates is that they're made up of carbon, hydrogen, and oxygen, but always in a ratio of 1 to 2 to 1. So you have your glucose, which is C6H12O6. And like all carbohydrates will have that ratio. So carbon, hydrogen, oxygen, always twice as much hydrogen as carbon or oxygen. I have glycosidic linkages here with no explanation. So I'm going to go ahead and explain what those are and why they're important. So glycosidic linkages are covalent bonds that hold the monomers together in a polysaccharide. So this right here is technically a disaccharide because there are 2 sugars, 2 monomers, mon meaning 1. So this would be your monomer or your monosaccharide in this case. And this is a disaccharide, but for this purpose, we're also going to pretend that it is a polysaccharide. So the glycosidic linkage is actually right here. And the glycosidic linkage is where you'll see a good example of dehydration synthesis. So because if you can imagine 2 of these, you have your hydroxide, your OH molecule, and when it bonds with another one, because as you can see, these are identical other than this, you have one hydroxide molecule here and one hydroxide molecule here, and you keep 1 oxygen, but you have between the 2 oxygen atoms and 2 hydrogen atoms. So if you take away 2 hydrogens and 1 oxygen, that's water. So dehydration synthesis takes away those extra atoms, allows the oxygens to bond covalently, and expels water. Dehydration synthesis. I think that's pretty cool. I don't know. Maybe I'm just a nerd. But so dehydration synthesis leads to glycosidic linkages, which are right here. That's what holds the carbohydrate polysaccharides together. And this is kind of also what I mean by big picture stuff in AP Bio. You're probably not going to get a multiple choice question or an FRQ question that specifically asks about glycosidic linkages. But I remember last year they actually showed us a polysaccharide, and they were like, how many covalent bonds or how many like bonds basically were within them, and you literally just counted them. So understanding things like this can be helpful. Understanding how they're formed, it's cool. It helps me connect and understand information. So I love it when things click for me. So I don't know about you. I also included this tidbit. This you probably won't need to know, but oligosaccharides can also covalently bond to proteins and lipids. So that's just another way that they all connect together. All right, so application to biological macromolecules, part 2, we have our lipids. I was not able to find the right picture. Like there was one specific one that I wanted that I just like couldn't find. So you're going to kind of have to bear with me, but I think I will be able to convey what I'm wanting to convey. So lipids are made up of mostly carbon, hydrogen, and oxygen. So there's that second show. Occasional phosphates. So think of, phospholipid, like a phospholipid bilayer. That's how I remember that. So the important thing with lipids and their bonding, what we just talked about to connect what I rambled on about for 15 minutes that you guys already knew, is that they store energy in their covalent bonds. So in their bonds between their carbon and then carbon and hydrogen. And again, there are no good examples in my picture here, which drives me crazy because I'm a visual person. But essentially, these legs right here is just a chain, or also like right here, is essentially just a chain of carbon, 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 and hydrogens on the end. And so that's where all that energy is stored within those covalent bonds. Excuse me while I sip some tea. Sorry. And this is also where we will see hydrophobic and hydrophilic aspect, that vocab that I talked about earlier, comes into play. So the phospholipid bilayer is made up of these phospholipids, and it's just incredibly important. So this section, this head, what we call it, is hydrophilic. It loves water. It is attracted to water. It basically lives in the water. And this is the hydrophobic tail. So essentially the bilayer, which I hope you're all familiar with, is essentially just like two layers of these phospholipids with the tails facing inside and the heads facing outside. And that is what allows, transportation into and out of the cell. And you'll also get a whole unit on cell transportation. So we're also not going to go into detail with that. But it is super important that part of it is hydrophobic and part of it is hydrophilic, and that's because it's insoluble to water. And, it's pretty important that they're insoluble to water because it is in water. So if it dissolved, then we just wouldn't have a cell membrane. So that would be bad. And those are the nonpolar covalent bonds that are allowing the lipids to be insoluble to water. And then we have so saturated and unsaturated fatty acids is another really important concept that is going to seem kind of not important, but it's one of those other ones that I think is really cool and really interesting. So in this phospholipid right here, we have our head, we have our tail. One of the tails is saturated and the other one is unsaturated. And they're both nonpolar, and because they're nonpolar, they are hydrophobic. So those two correlate as well. I don't know if I made that clear. And so the tail that is, okay, sorry, let me backtrack. The difference between saturated and unsaturated, and this is really where I wanted to try to get a diagram for you guys, just couldn't find one. Hopefully you'll see one eventually and it'll make sense to you. But a saturated fatty acid is just a chain of, it's basically a carbon chain, covalent bonds between all these carbons. And then they're also covalently bonding with hydrogens as well. So you have like a hydrogen down here, and your chain of carbons, and then above and below each carbon is covalently bonded to a hydrogen. So hopefully you're kind of with me and you can kind of visualize that. So that's what a saturated fatty acid is, because each carbon molecule is saturated or filled with a hydrogen bond. And then an unsaturated hydrocarbon chain, essentially what I just talked about, will contain one or more double bonds. So it kind of gets messed up if you have your chain of carbons and instead of being saturated with hydrogen bonds, not with hydrogen bonds, with covalent bonds to the hydrogen, it is double bonded to another carbon. And that's where you're going to find that little crooked tail. I know it's super duper hard to see, but if you actually look really closely in this picture, you can see that there's a double bond right there. I know it's super hard to see. It's just something that I like, I don't know, I can envision it. It helps me. I'm a visual person. And that's kind of what makes it crooked, because it's no longer saturated. It kind of sets everything off. So that's the main difference between saturated and unsaturated, is whether or not it is fully saturated with single bonds, or the unsaturated because it has one double bond. Sorry, just looking at my notes. And also another big, well, not big picture in terms of AP exam big picture, but big picture in terms of life, study of life. Animals use saturated fatty acids to serve as fats. So when the covalent bond between the carbon and hydrogen is broken, chemical energy is released, because like I said earlier, energy is stored in those bonds. And this diagram right here is a triglyceride. So it is a form of a lipid. It's not a phospholipid, very different lipids, but chemically similar and functionally similar as well. All right, so now I just have some review questions. You can feel free to answer these in your head, jot them down, answer them in the chat. And also, if you have any questions for me at this point between anything that I've talked about, or just about AP Bio as a class, as an exam, anything like that, feel free to ask them now in the chat or in the questions tab. And if you're confused about any of these questions, go ahead and ask as well. But to answer your other question, what do we need to know about water for AP Bio? Definitely know about adhesion and cohesion, because that's important with watering plants and how it goes up the xylem and such. Know about its polarity, which I kind of talked about, because that's really important. Definitely know what is and isn't soluble in water, like how I was talking about how phospholipids are not soluble in water. And just that kind of general idea. They're not going to test anything specific about water, but if you can explain why water has all these unique properties that is literally unique to water, because water is known for its cohesion and adhesion. And let me know if you're not familiar with those words as well, and we can go over that. But I will get back to that one, because I want to make sure that I am answering your question correctly. All right, so Zion asks, my teacher is teaching for the first year. How did you prepare yourself on your own? I was fortunate enough to have a teacher that is very experienced in AP Bio and very passionate about AP Bio. However, it was her first year with a new kind of curriculum, because my school used to make you take 3 semesters of AP Bio, and they just changed it to 2. And last year was the first year that they had done that. One thing that I really like to use is a website called albert.io. I don't know if you guys have heard of it, but it is a website that has a bunch of resources for multiple choice questions and FRQs. The only thing I will say is that FRQs are not graded for you, so you can practice doing them, but they won't give you a grade. It does cost money, but I found it super helpful, and I used it for both AP Euro last year and AP Bio. Other than that, Fiveable is a great resource to prepare on your own, because you're going to have so many streams, one-on-one time. It'll be great. Barron's books or any prep books will be good. Flashcards. AP Classroom. I'm actually not familiar with AP Classroom, but I think that sounds like a good resource. Let me know if that answered your question, but lots of practice tests helped me a lot. And just review videos. Oh, also Bozeman Science. He has great videos that I liked a lot. Crash Course has some. There's so many resources out there. All right, good. I'm glad that answered your question. All right, Dakota, so your other question, what are functional groups for lipids and carbohydrates? They don't have functional groups in the same way that amino acids and nucleotides would, if that's kind of what you're asking. Their job is more so to store energy than it is to do anything else, because proteins have functions to either break down or, catalyze with their enzymes, and they have their functions, and that's kind of what the functional groups do. If that answers your question. So I haven't really used the term functional groups for lipids and carbohydrates. So if that's not what you're asking, if you could rephrase, that would be great. But in terms of literal functional groups, I just haven't heard, I just don't think that they really have the same thing as proteins and nucleic acids, if you know what I mean. Okay, and then I'm gonna get down to this dehydration thing, because, and I'm gonna be honest, last year and even this year, because I took this class last year, I wasn't 100% on every single concept, and so dehydration synthesis did always confuse me. But yeah, so dehydration, water is a product of it, because you're taking away, like Peyton said in the chat, you are taking away the hydrogen and the oxygen from the two, we can go back to the slide even. I don't know if you're a visual person, I am. So here you're taking away this hydrogen, and then this hydrogen and oxygen, you're taking that away. So that's the dehydration part of it, and then you're releasing the water. So I know it kind of sounds weird because you want to think that it's using the water, but that's hydrolysis, because you're using the water to break it. And I know in my mind that's backwards, probably for yours as well. But yeah, I hope that answered your question. So yes, water is a product of the dehydration reaction. All right, I'm not seeing anything pop up. So that's all I have prepared for you guys today. Yep, so just make sure that you're following Fiveable, use it for your other AP classes. I know that you guys are probably taking multiple. It's a great resource. Yeah, just, good luck this year. You're going to do great. AP Bio is so much fun. I hope you guys enjoy it as much as I did. And, thanks for coming. Thanks for sticking it out with me. I hope you learned something.